I'll try the attendance quiz after I reboot, maybe in the second hour. And if that doesn't work in the second hour, just log into the site. But it's not, so my network card is not registering for some reason. Okay, so again, at the glacial pace that I'm proceeding, I'm not making much headway through the physiology, but I think we left off talking pretty much finishing a discussion, completing a discussion of membrane transport. So you should be very familiar now with the difference between passive transport, primary active transport, secondary active transport. You should understand the distinction between passive diffusion and passive transport. You should understand why in trans-epithelial transport you frequently have a secondary active transporter and a passive transporter. So all that should begin to make sense to you. We talked about the glucose transporters. We talked about the fact that by tradition, by custom, glucose transporters are designated GLUT, and those glucose transporters are all passive transporters. There's a little more complexity to them we need to discuss. But they're all passive with the exception of that S-loop. And I think there are about 3 variants of that, but we're really concerned about one variant of the S-loop. Good. For a number of reasons, usually for reasons related to the size of the substance that is either going to be taken into the cell or discharged from the cell, sometimes a transport mechanism, such as we've discussed, is inadequate. By that I mean that something that needs to be taken into the cell or removed from the cell is simply too large to be handled by one of the mechanisms we discussed thus far. So cells, again, this is not the only reason, but cells in general, if the particle is more than, let's say, 3/4 of a micron, that's the usual threshold you see, they'll use another process, an alternative process called endocytosis or exocytosis. And this is, I guess, the few things that I would have you keep in mind about these processes. Number one, they're energetically very expensive. They require lots of ATP, so make sure you understand these are not magical capabilities of the cell. They utilize a lot of energy, much more expensive than any of the other processes we've talked about. And then two, before we get into just the very simple discussion or the illustration of exocytosis and endocytosis, I want you to think about the nature of the cell. I realize we don't have time in this course to talk about cytoanatomy, and really all we've talked about at this point is the cell membrane. But you know that the cell membrane is largely a membrane, a protein-laden phospholipid bilayer. And there are other membranous structures inside the cell. We call them organelles. And in fact, in most eukaryotic cells, there is a vast endomembrane system. Perhaps from your biology or from your prior A&P, you remember things like the endoplasmic reticulum, right? The Golgi apparatus. Those are all part of the endomembrane system. All right, and the membranes that make up that endomembrane system are very similar. In fact, for all our purposes, identical to the plasma membrane, the cell membrane. All right. Now, as it turns out, and we'll talk more about this very shortly, things such as, I shouldn't say things, I guess, molecules such as proteins are synthesized and generated often in a portion of that endomembrane system. For example, ribosomes in the ER, in the endoplasmic reticulum, are frequently sites of synthesis for proteins. So we have proteins. In your opinion, are they primarily water soluble or fat soluble proteins? Anybody? Yeah, most of you say it all depends, but in general, they tend to be more water soluble. All right, so we have a paradox there. We're arguing that these water-soluble polymers of amino acids are being synthesized on a membrane, and so we'll analyze that problem in more detail later. All right, but what I want you to think about now is, let's say we have a cell then that is manufacturing protein, which you've told me is largely water-soluble. let's say that protein is a hormone or neurotransmitter that has to leave the cell. How is the cell, you're the engineer, you're the cellular engineer, how are we going to get that water, is that a problem at all? If I'm just if this wall up here is a membrane cranking out lots of proteins, and maybe that membrane down there is the cell membrane, will the proteins that I synthesize here simply go right through that wall down there to the blood, whatever? All right, so could anybody engineer a mechanism by which we can get the protein synthesized inside the cell to the blood?
Exocytosis.
Okay, so it says exocytosis, but all I have right now are a bunch of proteins. So what has to happen? What do we have to do with those proteins before we get them into the blood?
Package them.
Huh?
Package them.
Okay, good. Bartek says package them. So what do you mean by package them?
Vesicles.
Okay, excellent. You're going to put them in some type of vesicle. And my only point here, and we don't have time to get into this in great detail, is the package that Bartek's talking about is one of those endosomes that we remember, the lysosomes and endosomes. It wasn't a micelle. It was basically a little three-dimensional sphere with a hydrophilic core. And what's going to be in the it's a phospholipid sphere, but at the center of that sphere is a hydrophilic region that is containing what? Proteins. Containing proteins. And so now we have an intracellular vesicle. An intracellular vesicle. I've asked biochemists what that process is called, and they really don't. I said, would you accept vesiculation? And no chance. So we can call that vesiculation. That sounds silly to me. But anyway, we make up a vesicle. The vesicle is basically the package carrying the water-soluble molecule, in this case, such as a protein. Now, let's say this vesicle, for reasons, again, we don't have time to discuss in great detail, but these vesicles actually travel along a railroad system inside the cell. Isn't that what the railroad system is, or the interstate highway system? Well, not so much the Golgi. Now, I know what you're thinking, because in reality, once we fashion those vesicles, they go to the Golgi, and then, again, we don't have time to talk too much about that, where they get tagged later, get altered. But once they leave the Golgi, in most cells, in most eukaryotic cells, they hop on the interstate. And what's the interstate inside a cell? Anybody? Yeah, the cytoskeleton. I heard somebody say the cytoskeleton. And a lot of times it's fascinating to watch. If you ever go online and look at the animations, there are motor proteins inside the cell that will latch on to a big vesicle, and then they'll just start walking. It's like a man walking along the interstate. It's a dihedral protein on both ends. One end attaches like suckers to the vesicle, this massive glob, and then those little motor proteins just walks along. It's very fascinating to watch how it does it. There are other cytoskeletal components that do similar things. Well, my point is, now we have a mechanism by which we can get that water-soluble molecule packaged, we get it transported down to the plasma membrane, and then what happens? Because I want it out of the cell. I should be careful here what I say. One of the rules I have in this course is that neither you nor I should ever use the word it, because I never know what you or I am talking about when we use it. So in this case, I want the contents of the vesicle out of the cell. How is that achieved? Yeah. Exactly right. Exactly right. So what you sort of want to mentally conceptualize as you look at the illustrations, you can see the illustrations in the ensuing slides, is that phospholipid, that 3D, that three-dimensional phospholipid sphere simply melds, it simply joins with the plasma membrane down there. And in so doing, it involutes. It sort of discharges its contents into the ECF, into whatever is outside the cell. So the point here is that what I want, I guess the second thing I was shooting for here. First thing we said that exocytosis and endocytosis are energetically very expensive. Second thing is exocytosis is going to increase the surface area of the membrane. Everybody see why? Because the phospholipid sphere is now one with the cell membrane. What about endocytosis? Basically, in endocytosis, the membrane is going to invaginate, form a little pocket, and then bring the lysosome or endosome into the cell. Does the plasma membrane, does the cell membrane take a hit when that happens? It becomes smaller. So I just want you to see that there are some physical principles to keep in mind. The first principle relates to the conservation of energy. It takes a lot of energy to make and discharge vesicles, and it also takes raw materials, resources to discharge and incorporate these vesicles as well. So if we had to summarize the major reasons that cells undergo this process of endocytosis and exocytosis, again, I'm dramatically simplifying things. Number one, size dependent. If the particles, the molecules, or whatever in question are very large, conventional transport processes will not work. We need another step to use them. And then second, has to do with relative solubility, something we'll talk about. So as we've indicated, the interior of the cell is hydrophilic or hydrophobic? Yeah, generally hydrophilic. So that's the problem. If you discharge a lot of hydrophilic particles inside a cell, the problem is they get stuck there. They can't get out because the membrane is hydrophobic. All right. So one of the other reasons that these endosomes or lysosomes or vesicles are formed is to get hydrophilic particles through the cell membrane. Yeah. Maybe we can talk about your sources or whatever, but trafficking, in my reading and understanding, means something a little different. And we can talk about the as we talk about proteins and maybe protein hormones and how they're formed later. But trafficking is a larger process involving, for example, the Golgi and some of the other points that we'll talk about that later. If we compare sources and you have the better idea of trafficking, I'll certainly assent. By the way, a lot of you have extensive clinical experience, so feel free to, anytime we're talking about, especially in anatomy, if we're talking about some clinical or clinical topic or other topic upon which you have expertise, feel free to chime in. So you have a little animation here, simplistic illustrations of endocytosis on the left there and exocytosis on the right. And the key things to keep in mind is that under normal exocytosis, by the way, cells don't always work this way, but in normal exocytosis, only the contents of the vesicle are discharged. If the whole thing is discharged, we have another name for that, usually called a granule or something similar. So we have several illustrations here, and so when it says exocytosis is necessary for the process of secretion, secretion in this course is going to have relatively precise definitions. Unfortunately, they vary from one point in the course to the next. But in this context, we're talking about something leaving the cell in a rather precise action, a discharge of a substance from the cell. By a mechanism that usually conserves the membrane, conserves the phospholipid bilayer of the vesicle. This is an actual micrograph, and you can see changes in the cytoplasm that occur during endocytosis. We have, let me turn that over here. We have an invagination of the membrane. This is rather complex. We'll talk a little bit more about this in just a moment. And eventual formation of this phospholipid bilayer, which can then be taken into the cell and processed. Usually, when these things then wind up inside the cell following endocytosis, what organelle has the responsibility of inspecting and evaluating this package? Usually, anybody know? Kind of important for most cells, and you'll have a number of disease conditions in which that organelle is not doing its job properly, and it can be very, very problematic. What's the organelle that deals with, addresses, inspects? It's basically the virus detector. That's not a very good analogy. The what? Lymphocyte. Oh, lymphocyte. I've never heard that. That's fine. Oh, lymphocyte. Oh, no, so you're thinking of an immune cell. Yeah, we wouldn't have these, I guess one could say that the cell has its own immune system, but you wouldn't have, yeah. But you came close. I thought you were saying lymphocyte, because that gives you a clue. It's actually, yeah.
What about the lysosome?
The lysosome, yeah, good, exactly, the lysosome. So the lysosome is an intracellular organelle that uses hydrolytic enzymes to break down things like this, and it tends to, in most cases, and so it'll do the initial inspection and decide what needs to be done with this critter that's just moved into the cell. And then that, depending on a variety of circumstances, that phospholipid bilayer will be recycled.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
I want to make a few, I don't know if I can do this very well with my finger. So he described this process as, he called this endocytosis, but he said it is the nature of the substance that's taken into the cell that differentiates phagocytosis from endocytosis. The more modern, the more recent distinction is as follows. In endocytosis, there is, as you saw in the last portion of that animation, an invagination of the membrane, and following that, formation of the intracellular vesicle. In phagocytosis, there is an extrusion, as you see here, an extrusion of the membrane around the substance to be incorporated, and that's more typical of, say, a phagocyte. We'll look at a little video of phagocyte in just a moment. But that's the usual distinction. Otherwise, I don't have any problems there. And then the other thing, what was it? Something else he said bothered me. Well,
Okay.
yeah.
So is the distinction that in the
If the material
a vesicle is formed and endocytosis, the vesicle is not formed?
No, in both cases, an intracellular vesicle is formed. What I'm suggesting is that in traditional phagocytosis, as you see here, the cell is changing its morphology, it's changing its shape, and it's moving around it through cytoskeletal activity to actually incorporate the bacterium or whatever the bacillus is to be taken into the cell. And remember in our little illustrations of endocytosis, it's not that complicated. The cell doesn't extrude, it invaginates. And we'll look at another, in fact, in that clathrin-dependent endocytosis, the membrane invaginates, because that's what the clathrin does. But that's the distinction. I can't seem to, here we go. All right, so he's talking, there's a slide on this. This is sometimes called clathrin-dependent or receptor-dependent endocytosis. Now, first thing to keep in mind is most eukaryotic cells do not have a homogeneous cell membrane. They have the cell membrane, the plasma membrane of a eukaryotic cell, depending on a lot of factors, is very heterogeneous. It's mixed and varied. There's a lot of different surfaces. Some of it's covered with grass. Some of it's landscaped. You've got parks over here and so on. The simple analogy I'm trying to suggest is there are regions of a cell membrane that are specialized for specific activities. So they're going to be like landing strips, such as you see here, in which a region of the cell membrane will be covered with these receptors and clathrin molecules. And these clathrin molecules, as best we can figure, are actually responsible for pulling that membrane in by a mechanism that isn't well understood. And they do that, these little spheres that are intended to represent the clathrin, they do that when the purple thing here, which is the receptor, binds a specific ligand, right? So that's what we mean by receptor-mediated or clathrin-dependent endocytosis. It's a specialized region of the membrane that has both receptors and clathrin molecules. And the other thing to keep in mind is a little bit different from our discussion of a ligand-gated receptor. In this case, what's being taken into the cell?
Bacterium.
Well, it could be you're exactly right, but in this simplistic illustration, what color is the thing that is being taken into the cell? I guess what I know that's a silly way to ask the question. The cell is undergoing this process in order to bring one of these things into the cell, which is the thing that's most important, that it's most concerned about. The ligand, the red thing. And everything else is recycled. Everything else except for the red thing up there, which is the ligand. It could be an LDL, it could be any number of things. It's taken into the cell. A vesicle is formed. There's a little illustration of this that follows. And then again, it's usually sent off to a lysosome for processing and deliberation and judicial proceedings. Yes?
So basically, in endocytosis that's receptor-mediated, there's no cytoskeletal rearrangement?
I think, well, I wouldn't say there's no cytoskeletal rearrangement. It's just not the basis for the incorporation of the foreign substance into the cell. That's more typical of phagocytosis. And the last thing I would say about this, and again, you have I think that was all I was quibbling about with that. Yeah, here's the illustration. Virtually I get careful when I tend to be hyperbolic. Most eukaryotic cells have these specialized clathrin-coated regions on the membrane. And most cells, even if they don't use clathrin, have some other type of receptor-dependent mechanism for bringing things into the cell. So in this way, what we're saying is the cell is sensing its environment and responding accordingly.
Is all endocytosis receptor-mediated?
No, the question was, is all endocytosis receptor-mediated? Probably not. I guess the only point I'm making is that virtually all cells have this, so it must be important. It must be an essential component of their metabolism and physiology. Now, we mentioned phagocytosis, and you heard my little distinction there. So what are we looking at here? Of course, you know what these things are? Those are going to be erythrocytes. And so what is this then? It's a phagocyte. And what is it doing?
Chasing
Yeah,
bacteria.
what's that little critter there? And how, isn't that, there you go.
Wow. That's awesome.
Now my question is, how did it know, how is it able to hunt that sucker? By what mechanism? I mean, this is probably going to give you nightmares. That thing's just minding its own business. And here comes this massive blob. Isn't there a movie recently called The Blob? It's probably leaving a little chemical trail, and that phagocyte is probably sensing that in some way. And the mechanism by which that macrophage engulfed the coci, or whatever that was, would be representative, characteristic more of phagocytosis than endocytosis.
Is phagocytosis a form of endocytosis in this lab?
You're trying to pin me into corners. I take a very liberal view of things. I'm just kind of free floating. I would discourage you from trying to be too precise. We're going to have that problem over and over again. And I would say, in many cases, there is no precise distinction. But often my emphasis is pedagogical or didactic. So in here, I'm sort of artificially to some extent, imposing this distinction between endocytosis and phagocytosis. So for our purposes, let's say they're distinct based upon mechanism. But in philosophic way, yeah, I think they're both forms of bringing something into a cell, which is what endocytosis is. I don't think there's anything new in this slide. We talked a little bit about secretion. Secretion is a form of exocytosis, or often can involve exocytosis, and we see here we have a cell. The other thing is the cytoplasm of the cell. Here's the cell membrane over here. And then, as was pointed out earlier, this is part of the endomembrane system called the Golgi apparatus, or Golgi body. And especially in cells that are specialized for exocytosis or secretion, the Golgi, part of that endomembrane system we talked about before, is fairly close to, it's fairly near the plasma membrane, because as you can see here, one of its jobs is to evolve or generate finished vesicles that carry something that is ready for export, ready for shipping and export off to some other distant land. And so now we have these endocells. They're calling them here a secretory vesicle, and by now I guess that's the term we should use. As we'll learn later, vesicles that leave the ER and end up in the Golgi are usually called transfer or transport vesicles. We use the term secretory vesicle to refer to a vesicle that the contents of which are going to be exocytosed, going to be secreted. So the only other significance of this image is that we are differentiating what's called constitutive secretion from regulated secretion. Just as we found, we talked about ligand-dependent endocytosis earlier. Constitutive in this class will mean normal, ongoing, regular. So most cells in your body have to undergo constitutive secretion for no other reason to get rid of metabolic waste. All right, so constitutive secretion is normal, ongoing, and we'll use the term in that way. By contrast, regulated secretion means that the contents of this secretory vesicle are only discharged, are only exocytosed when what happens? A membrane receptor binds a ligand of some sort. This could be a hormone, this could be some other signal molecule. And then through a process we'll talk about later, through a process called second messenger signal transduction, which you're going to enjoy thoroughly, that will signal this whole assemblage. We talked about the motor proteins and all that in the interstate highway system. That will trigger exocytosis, as you see it here. So constitutive, normal, doesn't require any signaling. If it's regulated, the regulation here implies usually binding of some type of extracellular hormone or something to trigger this. Now this should look familiar to you. This is something we talked about before. Can somebody hold forth on this for a few minutes and give me a break? What are we looking at here? What is that yellow thing? Oh, good. So it's an epithelial cell, could well be in the intestine. How do we know which side faces the lumen and which side faces the blood? All right, the ruffled, that's the tradition. So the lower top is the luminal surface. That's the surface facing the interior of the intestine. We'll use that example. And the ISF and blood would be down here, the ECF, the ISF portion of the ECF, and eventually the blood. So what are we looking at? What are all those things in the membrane? What's that thing up there on the apical surface of the membrane? We have this. Okay, excellent. That's a very sophisticated, fancy terminology. Bob says S-glut. What is an S-glut? Secondary active glucose transporter. Good. Is it a symport or any port in this case? Okay, good, symport. And so it's acting to increase the concentration of glucose inside the cell. What is this thing? That's the sodium-potassium pump, all right? And we know that this is necessary in order for that thing to do its job. Therefore, we say that that's why that secondary active transporter on the apical surface is sodium dependent. This is the sodium that we're talking about here. And then finally, what's this? Right, that's a passive glucose transporter. It just allows the glucose to pass through the membrane according to its concentration gradient. Presumably, the concentration is higher inside the cell than outside because of the activity of the secondary active transporter. And these are all over the place again. I just want to remind you of the importance of this arrangement, this organization of transporters in the membrane. Of course, there is I guess it's still a debate going on about the significance of fructose. We said that in the intestines, glucose uptake in the intestines is sodium dependent. That's predominantly true. There's some exceptions. And you know that there are at least 4 different passive glucose transformers. But perhaps what you didn't know is that in your intestinal epithelial cells, there are also specialized transporters for another sugar, fructose. So you have, most of the epithelial cells have these specialized transporters that bring fructose in. They work by different mechanisms, what we've discussed. And so the body takes in, the GI tract takes in fructose rather liberally, takes it in rather freely. And so have you been keeping up with this debate? When you go to the grocery store, do you look for bread? Can you find anything that doesn't have fructose in it?
It's hard.
And so I think fructose is in the wax they put on the floor. Fructose is in the air we breathe. It's just everywhere these days. It's in all our drinks pretty much. Of course, now that people are alerted to this, you'll sometimes see on the label, on the bread label, no high fructose corn syrup, and so on. So I just want to alert you to the fact that maybe it's something you want to look into, investigate if you haven't done so already. And the relevance in this context is that, once again, the GI epithelial cells have specialized fructose transporters. We talked about, we need to say a little bit about this as we move to our discussion of osmosis and tonicity. We talked about total body water. We know the 60-40-20 rule. We don't need to go over that again. But what I want you to think about a little is just a couple of principles involving what's called the measurement of the fluid compartments of the body. Perhaps this illustration is all we need to look at. You know that if you introduce a fixed amount of something into a volume of fluid, that will result in a determinable concentration. If we increase the volume of fluid into which we introduce that fixed amount of something, does the concentration go up or down? Down. So very simple. So, depending upon which type of substance you introduce into the body, and depending upon its relative permeability, its ability to pass through, as you can see in the one case, the blood, get out of the blood supply, or make its way through the entire ECF, from both the plasma and the ISF, or finally, in the case of what is called antipyrine, which I think is an analgesic or something, they use that for, what do you use it for? Oh, otitis, earaches, I think it's called, they use it for anymore. Or tritiated water, which of course is radioactive, I assume they use it in relatively low concentrations. But you can see, depending on whether we use Evans blue, inulin, or let's say antipyrine, the volume, the body water volume is increasing. Therefore, you would expect the concentration of the substance introduced to go down. And if you know the relationship between those 3 compartments, as we talked about before, you know the relationship between the 2-to-1 relationship between the ICF and the ECF and so on, you should be able then to make a determination about the volume of fluid in the plasma, the plasma, volume of plasma, the volume of the entire ECF, or TBW, the entire volume of total of the water in the body. So that's all this is talking about. I just want you to understand that there is a reason that you should know about these compartments and how the relative volume of each can be determined if you use substances of varying permeability. All right, so we talked about diffusion to continue our discussion of general principles that underlie the dynamics and metabolism and physics of cell membranes. Let's talk a little bit about osmosis. I think all of you know the definition of osmosis. There are some potentially dangerous definitions that I sometimes see in textbooks. And virtually all definitions that you will see in a biology or physiology textbook are incorrect. Fortunately, it doesn't matter because we're going to define osmosis in a relatively simple way that avoids the problems of those definitions. We will argue that osmosis is a physical process whereby water, we're only talking about water here, moves across a selective membrane, usually a cell membrane of some type. We've already talked about various phospholipid bilayers and where they're encountered. Movement of water across a saran wrap or a cellophane type membrane, biological membrane. And then here's the key part, number 3 there, toward total higher particle concentration. There's a key word missing there. That's why I stumbled. You need to add total in front of particle. Total particle concentration, very important. Because osmosis doesn't care what kind of particle we're talking about. If you're talking about proteins or ions, it doesn't matter. I will say that osmosis is not the diffusion of water. They're actually not comparable. But as I say, it won't matter for our purposes. Let's just use this different definition and not be concerned about things such as water concentration. That will only lead you astray. So just learn that definition very thoroughly. We have a beaker here. We have at the center of the beaker some type of, let's say it's some type of, cell, membranous chamber that's encapsulated by this differentially permeable membrane, and it has a 360 grams per liter sucrose concentration inside it. Peripheral to that cell, still inside the beaker, we have a concentration of glucose which is half that. Now, what's the illustration telling us? What's the water molecule doing as a consequence? Moving toward higher total particle concentration. So make sure you understand osmosis is that simple. And it's determined the movement of the water, once again, is determined exclusively with respect to, number 1, the relative concentration of all particles, independent of their identity. And number 2, complicates things a little bit, the permeability of the membrane to water. We'll assume that all of the membranes that we're discussing are permeable to water, because that would be, I don't think I want to go down that road right now. This is not a terribly important distinction in here, because in life sciences we normally deal with osmolality. But osmolality is concentration by mass. Osmolarity is concentration by volume. Yes?
Would it matter if the particles are charged or not, or just if the concentration is higher?
Strictly speaking, it will not matter at all, because if we're, let me try to, this is part of the discussion I was trying to avoid. The charge would affect the ability of the particle to pass through the membrane with respect to diffusion. But with a very purist approach to osmosis, it would not, because ions, this osmosis applies again to ions, proteins, and so on. So don't confuse Fick's law with our simple discussion of osmosis. Yes?
I just want to make sure I have this right. Osmolality is a concentration according to mass.
Concentration by mass. So we would talk about the total number of particles by kilogram of water, right? Whereas if we're describing osmolarity, or we're expressing the concentration in osmolarity, it would be total number of particles per liter of water. Now, why would that make a difference? As I say, normally in the life sciences you use osmolality, but why would that make a difference? Are all of the solutions in your body, all of the fluids in your body of equal density? What's density? Specific gravity or density? Yes, mass per unit volume. Yes. So for example, would blood have the same specific gravity or density as pure water? Probably not, it wouldn't. What about cerebrospinal fluid? It's not even going to have the same density as blood. And then you have other fluids in the body and so on. So, as I say, for reasons we won't get into right now, usually physiological fluids, IVs and so on, are usually expressed with respect to mass. So it's not important here. This is not a biochemistry course, but that's the distinction. You should know the distinction in these terms, osmolality versus osmolarity. And you should know life sciences usually use osmolality, but I'm not going to ask you to do any calculations on that basis. And so, in the silly animation you see here, we're just saying as the particle concentration on the left side increases, the volume of water also increases because it's passing from the right. See, so this is going to be important. Osmosis. Osmosis is basically a statement of concentration. Osmolality is basically a statement of concentration. It's just talking about the total number of particles. If we have a higher total number of particles on one side of the membrane, that's going to result in water moving toward that size. And then this gets us to another term we'll talk about in just a moment. We have a change in the volume of water across the membrane. And that's why osmosis is important for you as a clinician, because you need to anticipate the consequences of providing a patient with a oral therapy or IV. You need to understand how that's going to affect the movement of fluid across the compartments. Yeah.
Theoretically, if you had a bunch of particles on one side of the membrane and no particles on one side, would all of the water pass through that membrane to the
I'm not sure that I entertain theoretical questions in here. No, it wouldn't happen for a reason we'll talk about in a moment, but it would have to do with something called osmotic pressure. And since you asked the question, it may very well be the next slide. No, it's not. But understand that all of these concepts are a little more complicated than we have time to address in here. We've already pointed out, for example, that in a true biological context is often the presence of these aquifers, these water channels that determine relative permeability. Now, in our discussion of aquifers previously, we said that water does have the ability to pass through membranes if two things are present, even to some extent independent of the aquifer concentration. If the total particle difference is so great that there's something called bulk flow occurs, it's basically the water molecules, the physical forces overwhelm the membrane. The portal biological membrane doesn't have the ability to withstand the tidal rush, the rush of water across the membrane. But otherwise, I just want you to understand that in reality, when we talk about renal regulation of fluid volume or in the various organ systems of the body, those aquifers are extremely important in determining the relative permeability of those membranes to the movement of water. So two things before this complication gets a little more before the discussion becomes a little more complicated. We've said that osmosis is the movement of water across the membrane toward higher total particle concentration, and that osmolality, we'll just use that word for now since I happen to remember it. Osmolality is simply a statement of concentration. How would somebody define osmolality? I'm not concerned about the liter or kilogram. What type of statement of concentration is it? Okay, good, you're right, you're exactly right. That's not what I'm focusing on. I just want you to understand, because this is going to be important in just a moment. Osmolality is a concentration, and I'm concerned about what's in that numerator. What's in the numerator? No, that's in the denominator. Particles, not just particles, total number of particles regardless of what they are. So make sure you understand that when we're talking about, I should have said osmolality or osmolarity. It's just a statement of concentration. It's a number on a bottle. It's like the number on the room out here. It just stipulates the concentration, the total number of particles in that volume or mass of fluid. And that's all it is. Don't impute to the term osmolality any other meaning. It's just a number on a bag or a number on a bottle or whatever. So with that in mind, looking at these terms, because I want to try to head off a problem we're likely to encounter, an isoosmotic solution would be what? How would you characterize it? First of all, isoosmotic, iso meaning the same as what? Same as what?
Equal particles.
Okay, so you're exactly right, the same number of particles, but I still ask you, if I hand you a solution and I say, hey, this is great stuff, it's isoosmotic. Everybody's impressed. Isoosmotic solution, I'm telling you that my bottle of solution is comparable to something, what?
Isn't it the same as like the concentration of your blood?
Yeah, I think that's pretty good, yeah, the ECF. So that's another thing that you should keep in mind, that unless you're given some other information to suggest otherwise, especially as a PA, when you see these terms in the clinical literature, isoosmotic or hypoosmotic or hyperosmotic, the reference, the comparison is generally the ECF for the blood. Yeah. Okay. And we'll talk a little more about that in just a moment. But that's how it is. Mm-hmm. So with an isoosmotic solution, what conclusion would you derive from that word? That the solution has the same total particle concentration as the ECF, or at least approximately the same. That's it, nothing more. Period. If the solution I give you in the bottle, for example, go down, you can't really see it because I didn't use very high. This is magnesium citrate. Anybody had any experience with magnesium citrate? Yeah. So let's not tell the others. What I would encourage you to do on the way home this afternoon, grab some. It's tasty, just a light fluid. And I think a whole bottle is about the right amount. So let's think about the bottle. What would be the consequence of that? Anybody know?
Yeah,
you're going to be in the toilet. You're going to be powdered for a while. But the mechanism is a little complicated. But basically, by introducing that hyperosmotic solution to the colon, that induces rather effective diarrhea. Is what you get. But the trigger here is the higher total particle concentration. So do you have any, before we take a break, are there any questions then about what we mean by isoosmotic, hyperosmotic, and hypoosmotic? These are simple adjectives to describe the number of total particles relative, for now, to the blood, or the ECF. All right, let's take a break.